following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
So what this slide does, it gives you a nice diagrammatic representation of the transcription of a gene. Now, I'm talking about transcription specifically in eukaryotes because remember, as I said, prokaryotic organisms do not need any assistance. They do not need any help when it comes to binding to DNA to begin transcription, but eukaryotic cells do. So like I said, the first step in the transcription process is initiation. And it just basically involves the binding of RNA polymerase. And as you can see, that black, that gray, that oval-shaped sphere right there is what I have used to represent RNA polymerase. And transcription begins when that RNA polymerase actually binds to the promoter sequence. As you can see, that's the promoter sequence right there to begin transcription. So once RNA polymerase binds to the promoter, it causes it to grip tightly onto DNA. And then the RNA polymerase starts to move along the strand that it wants to transcribe. And it doesn't start making the actual mRNA transcript until it gets to a specific region or a specific sequence that we call the start point. So transcription doesn't actually happen as soon as RNA polymerase II binds to the promoter. It actually starts, or the actual mRNA actually starts being formed, being synthesized, once that RNA polymerase moves downstream and passes or gets to the start point. Now, elongation is basically the movement of that polymerase downstream, down the DNA molecule, unwinding it as it goes, and elongating the RNA transcript in the 5' to the 3' direction. So what I want to point out in this case, and what's also very important, is as you can see in the second step, elongation right here, we have that being the 5' end of that newly synthesized mRNA strand, that red mRNA strand that's being synthesized, and the other end being the 3'. So the strand is synthesized from the 5 to the 3' direction. That's an extremely important fact, one of the most important that you should remember as far as transcription is concerned. Okay. Now, another thing that I need to mention as far as elongation is concerned is that the RNA is typically very efficient. So you will find it transcribing approximately maybe 40 nucleotides per second in the case of eukaryotes. And like I said, you can have several RNA polymerases binding to a single gene or the same gene, and all of them making large amounts of mRNA. So you can have large amounts of mRNA being produced from a very small segment of DNA. Now, in the third and final step, so that is elongation that we talked about. Now, in the third and final step of transcription, which is termination, there are a few important distinctions, and I'm talking about prokaryotes versus eukaryotes. Okay? In bacterial cells or in prokaryotic cells, once RNA passes a sequence that we call the terminator sequence, once that terminated sequence is transcribed, it'll make the bacterial RNA polymerase fall off and release the RNA transcript. So the termination process in prokaryotes is extremely straightforward. Once the terminator sequence is transcribed, RNA falls off, the transcript is released. In eukaryotic cells, on the other hand, what happens is that the RNA polymerase will transcribe a region that we refer to as the polyadenylation signal sequence. The polyadenylation signal sequence. Now, once this sequence has been transcribed by RNA polymerase, about 10 to 35 nucleotides downstream of that sequence, proteins that are associated with the growing mRNA transcript will then cut it free from the polymerase and release it as what we call pre-mRNA. So why I'm calling it pre-mRNA is because in eukaryotic cells, mRNA has to go additional processing before it's converted into mature mRNA that can actually be used in protein synthesis. So in the case of prokaryotes, we have the transcription of the terminator sequence that ends transcription, terminates transcription. And in eukaryotes, we have the transcription of the polyadenylation signal sequence on the length of DNA, which codes for the polyadenylation signal, which triggers or signals for transcription to end or for transcription to stop. So that's transcription in a nutshell. Okay, now I recognize the fact that there are some things I didn't talk about on the slide. More specifically, I didn't really talk about the transcription factors, which were involved in the initiation of transcription in eukaryotes. Now that was deliberate because we're going to talk about them at length in the next slide.